Today, we are going to cover Chapter 4, Sensation and Perception, and I chose to really focus on this idea of the skin senses, so touch, pressure, temperature, pain. Another word for this is called proprioception, um, and in a nutshell, what you see, or sort of what happens, is that the nerve cells the nerve cells, so the afferent cells, are located at different depths throughout the skin. So if you have a third-degree burn, it's going to activate many different levels of skin receptor cells, which is going to send a message to the brain, which is going to then activate the motor neurons, or the efferent neurons, and cause a change in your behavior, which would be to pull your hand back. But another area that is actually very interesting, and there's much more sort of research looking at what is pain. And I think that there's one particular area that that's interest to me is this idea of a phantom limb. So how our brain programs information about what we have on our bodies, as well as sends information out to our bodies. So the phantom limb is an interesting scenario where people are feeling pain in a leg that they don't have. And so this clip is going to sort of talk about the research, but what I think is really fascinating is that the research is showing that it's not just some crazy thought that this person may have, but it truly may be that our brain has rewired, that it's the plasticity, or that the brain still in the brain itself still perceives that there is a leg, and so it's still sending information to that leg even if that leg doesn't exist. So I will let the experts speak to this, and I'll pull the video up and I'll highlight a few points.
Imagine losing a limb through surgery or accident. Then imagine experiencing genuine pain in the limb that's no longer there.
For somewhere between 50 and 80% of people who have undergone an amputation, this is a very real experience. Known as phantom limb disorder, phantom limb pain, or simply PLP, this post-op phenomenon occurs most often in people who lose an arm or a leg. Phantom pain may also follow the removal of other body parts, including the breast, penis, or eye. No matter what body part is amputated, the condition manifests as recurrent pain in that spot. Some people also report other sensations like cramping and tingling in the phantom part. Doctors once thought PLP was a psychological condition brought on by a patient's grief over the lost limb or body part. They now believe that these feelings more likely stem from the central nervous system. Researchers think that if this is true, PLP may be explained by confused nerve endings at the site of the amputation, which continue to send pain signals to the brain even after the limb is removed. Or they think it could be due to the brain rerouting the missing limb's sensory input to another area of the body, such as from a missing foot to a real hip. In a case like this, any stimulation to the hip would also be felt in the phantom foot. Because its cause is unclear, phantom limb pain is difficult to treat. Doctors may prescribe antidepressants like Elavil and anticonvulsants like Topamax or Neurontin, which minimize the transmission of pain signals. Experimental therapies may help treat PLP. common options include acupuncture and transcutaneous electrical nerve stimulation, a procedure where weak electrical currents are sent to the area of discomfort in the hopes of dulling pain signals. Hypnosis, massage, and relaxation techniques are also options that may help ease PLP. Despite the existence of these treatment options, a recent survey found that some 53% of amputation patients don't seek out help for the problem. Don't be among them. If you're about to undergo an amputation or you've recently experienced one, talk to your doctor about phantom limb pain.
So, what I think is particularly interesting about this is one, they had wonderful visuals of the synapse and communication with neurons, is that our brain does not always, the function does not always serve a purpose. And what I mean to clarify that part is that if the brain is sending pain signals to an area that no longer physically exists, then the brain, the central nervous system, is not always aware of the function on the other somatic parts of the body. So I think it's just interesting to think about how the programming of the brain can be malfunctioned as well as functioning for the good of the body. The other part that I think is very interesting is how our senses interact. So one area the book talks about is synesthesia, and I found a video that I think explains what the experience is to have one sensation evoke another. So I will put that video on now.
Imagine a world in which you see numbers and letters as colored, even though they're printed in black, in which music or voices trigger a swirl of moving colored shapes, in which words and names fill your mouth with unusual flavors. Jail tastes like cold, hard bacon, while Derek tastes like earwax. Welcome to synesthesia, the neurological phenomenon that couples two or more senses in 4% of the population. A synesthete might not only hear my voice, but also see it, taste it, or feel it as a physical touch. Sharing the same root with anesthesia, meaning no sensation, synesthesia means joint sensation. Having one type, such as colored hearing, gives you a 50% chance of having a second, third, or fourth type. 1 in 90 among us experience graphemes, the written elements of language like letters, numerals, and punctuation marks, as saturated with color. Some even have gender or personality. For Gail, 3 is athletic and sporty. 9 is a vain, elitist girl. By contrast, the sound units of language, or phonemes, trigger synesthetic tastes. For James, college tastes like sausage, as does message and similar words with the "age" ending. Synesthesia is a trait, like having blue eyes, rather than a disorder, because there's nothing wrong. In fact, all the extra hooks endow synesthetes with superior memories. For example, a girl runs into someone she met long ago. Let's see, she had a green name. Deezer Green, Deborah, Darby, Dorothy, Denise. Yes, her name is Denise. Once established in childhood, pairings remain fixed for life. Synesthetes inherit a biological propensity for hyperconnecting brain neurons, but then must be exposed to cultural artifacts such as calendars, food names, and alphabets. The amazing thing is that a single nucleotide change in the sequence of one's DNA alters perception. In this way, synesthesia provides a path to understanding subjective differences, how two people can see the same thing differently. Take Sean, who prefers blue-tasting foods such as milk, oranges, and spinach. The gene heightens normally occurring connections between the taste area in his frontal lobe and the color area further back. But suppose in someone else that the gene acted in non-sensory areas. You'd then have the ability to link seemingly unrelated things, which is the definition of metaphor, seeing the similar in the dissimilar. Not surprisingly, synesthesia is more common in artists who excel at making metaphors, like novelist Vladimir Nabokov, painter David Hockney, and composers Billy Joel and Lady Gaga. But why do the rest of us non-synesthetes understand metaphors like sharp cheese or sweet person? It so happens that sight, sound, and movement already map to one another so closely that even bad ventriloquists convince us that the dummy is talking. Movies likewise convince us that the sound is coming from the actors' mouths rather than surrounding speakers. So, inwardly, we're all synesthetes, outwardly unaware of the perceptual couplings happening all the time. Crosstalk in the brain is the rule, not the exception. And that sounds like a sweet deal to me.
So I'll end today's lecture with one more example about how our senses interact, and that's through multimodal perception. And I've talked a lot about the study that I did, and so I've put some figures in from the publication itself. So what we were looking for, to give you a little bit of a reminder, is how do infants integrate and coordinate information from faces and emotions. So this is the title of the paper, which is Building Biases in Infancy, the Influence of Race on Face and Voice Emotion Matching. The logo is my maiden name, so that's me. So what we found was when we paired two faces, happy faces or sad faces, so we collapsed across the variable of emotion, that 5-month-olds and 5-month-olds looked at the new face. So we had them look at a face until it was boring, and then we paired that same face with the new face. Looked at the novel face greater than 50% of the time. So they were able to perceive that that was a new face. But what we found also was for the 9-month-olds, they were only able to visually discriminate between the same race faces, so the Caucasian faces. So as you can see here, they looked longer to the novel Caucasian face, but did not look longer to the novel African American face. So then we took this one step further. And what this is, so we've talked about EEG, and in the past lecture for chapter 3, I talked about event-related potentials. And so this is a map of electrodes that are measuring neural signals that rise to the skull. So what areas we focused on were in the frontal lobe, right? And in the occipital lobe, because we wanted to look at visual processing as well as this cognitive part. So how did we do that? So what we did was these babies wore this funny hat that corresponded, and there were 128 electrodes on this this net, as we call it. And while they were wearing this, we recorded the neural activity in response to a particular stimuli. So what they saw was this sort of neutral patterned background, and we put the cross in the middle so that they would attend to it. They heard a sound for 800 milliseconds. They saw that same pattern. They saw this slide again, or they saw that same picture again. And then there was a face, and it was either Caucasian or African American, and it was either congruent or not congruent, right? So as you can see, these, in the congruent condition, they heard a happy sound and saw a happy face. In the incongruent condition, they heard a sad sound, and saw a happy face. And then in between, So this right here is one trial. So in between each trial, there was about a second break. So when we look at the frontal region, we looked at 5-month-olds, and so we collapsed we looked at 5-month-old responses to Caucasian faces and African American faces. Now here is where the important part is about event-related potentials, is that we looked at and were able to average the response to a Caucasian face that is congruent with the sound, so that it was one, because that's one type of trial, it's one event. And we were able to average all those trials together so that we could get a waveform. And this waveform is the average of all the babies that had usable data. And so what you can see here is that in the frontal region, we really don't see any statistical difference, and we see a similar pattern of neural response in regards to when the 5-month-olds see Caucasian faces and African American faces. However, when you look at 9-month-olds, what you do see, and this is in the occipital lobe, so when we're looking at visual processing, is that you do see a difference in the visual processing for Caucasian faces. So they are able to detect what one could say that this data suggests that they are able to detect congruent sounds, so happy emotion sounds, and whether they match with the emotional face. However, what's really important here is that they can only do it for Caucasian faces, but not for African American faces. So again, and when you just take one variable, which is the race of the face, you can see that the neural response in the occipital lobe is larger for Caucasian faces than African American faces. So that's the end of the lecture portion here. I just wanted to touch upon a few class things. So the rubric is posted. A student had a question about highlighted versus italicized. All that matters to me is that you include a couple of terms that are not pre-highlighted or pre-italicized for you. So it's just another term that stands out to you that the chapter has not identified as a study term. For the glossary, the submission format, please do one journal entry per chapter. So that means that when you say chapter 1, it should only have terms from chapter 1. It makes grading for me much easier. The citation and references, I've gotten a lot of questions about this, so I wanted to post an example. The brain is an organ that controls our body and has 4 lobes. Feldman, 2015. So I have the author and the year. If you look on the home page, course resources, there is APA citation. You can Google it. Whatever source you need is fine, but I just want to be explicitly clear. As stated in the netiquette, the syllabus, and in all of my lectures, that APA format and learning that format is absolutely necessary. The survey results, I just wanted to say thank you so much for everyone who anonymously gave me feedback about the lectures. This lecture is a result of that, and for office hours, I will get back to you soon about that. So thank you so much for your time, and take care.